So first off, I'm going to introduce who we are as Cogist, and then I'm going to introduce Dr. Urai, and then going to talk about the details of the event, and then we're going to proceed with the talk. All right. So once again, hello everyone. This is Nestiham from Cogist, which is a community where students and professionals from various fields of study gather around to make resources based on cognitive sciences more accessible and provide students a way of communication with the various events we hold throughout the year, such as Academic Cognition, which is a series of talks preferably given by undergrads and grad students who are relatively less recognized. Book Cognition, that provides an opportunity for professors and students to exchange ideas revolved around a book of choice for several weeks, and Growing Up in Science, where professors share their lifelong experiences about the academic life. You can follow our accounts on Instagram, LinkedIn, and Twitter, and you can also support us from the patreon.com, and we would truly appreciate your support. Today's event, named the Cognitive Webinar, is designed to be online and held entirely in English, aiming for an international audience. We would like to provide a platform not just for science communication, but also interaction of academics from all over the world. Today, we're going to listen to Dr. Anne Urai, who is a cognitive neuroscientist and an assistant professor at Leiden University's Cognitive Psychology Unit. She did her doctoral degree with Tobias Donner at the University Medical Center, Hamburg-Eppendorf, and completed her postdoc in Anne Churchland's lab at Cold Spring Harbor in New York. Her research focuses on how the brain transforms sensory information into useful decisions and how decisions can change with experience. Today, we're going to listen to her talk entitled Choice History Bias as a Window into Cognition and Neural Circuits. Her talk will last about 40 to 50 minutes, and then we will give a 5-minute break. After the short break, we will proceed with the Q&A session, which will also last about 30 minutes approximately. Before we proceed, I would like to emphasize the fact that we would like our attendees to ask their questions with their microphones and video cameras open if possible, but it is not a requirement. You can also ask questions through the Zoom chat as well. So Dr. Urai, if you're ready, you can go ahead.
Yes, thank you for that nice introduction and for having me. It's nice to see so many people here, some familiar names and some new. If you feel, like anything is unclear, you have some questions during the talk, please just interrupt me. You can raise your hand or type it in the chat. I would much rather have a little bit of an interaction during the talk as opposed to keeping everything afterwards. I find myself when I attend talks that it's easier to ask a question straight away than to keep it in my working memory towards the end. So, feel invited to do so. All right, yeah. I won't read you the title of the talk again, but I'll just start by going back to the time when I began my PhD in cognitive neuroscience quite a few years ago now. And the broad idea in this kind of subfield of study of perceptual decision-making is that we understand how we take noisy sensory information that is sometimes difficult to judge and make a decision about what we perceive there to be in the world. And of course, these decisions are then crucial to guide useful actions. Now, one kind of real life example of this scenario is that you might be walking in the park and see this animal in the bushes. It may be a little bit foggy as in this picture. So it's difficult to know if this is just a local neighborhood cat, in which case you can walk ahead, or that this could be an escaped tiger from the zoo. And of course, in this particular example, this judgment about the visual input that you have to make has a big consequence for the action that you would take, in this case, perhaps running for your life. Now, in the lab, we normally use studies that are a little bit kind of less exciting, but allow us more control over really studying the input-output between sensory information and these perceptual judgments. So for instance, you may have heard about this task called the random dot motion task, which I'm showing here on the screen. It's normally pretty hard to see it in this Zoom, on the screen through Zoom, but what you may see is that there are periods when these flickering dots are all moving together into in this case, the bottom right corner of the screen. So we use these types of tasks to very precisely alter the sensory information that we give people to then make a choice about what they see. And we can then use mathematical models to study this process that underlies this decision-making transformation. And when I started my PhD, I was thinking a lot about different types of models to explain or capture this process. And what we would then do in the lab is ask people to come in, put them in front of the screen, and give them a lot of these stimuli, one after the other, to study this process. For instance, this is just a sequence of trials that we gave one example person in a data set from a paper that I worked on with Anke Braun. And we gave people these stimuli where the dots in these flickering patterns could move either upwards or downwards with a varying intensity. And most of the models that then capture this process of this sensory transformation rely on a core assumption, which is that the decisions that you make one after the other are actually independent of each other. And that each time you kind of start fresh, you take all the sensory input that you can get and you use it to make your decision. All is well. But when we look at the responses that people give on these types of tasks, we actually see that this assumption of independence is very often violated. So in this particular case, you see that this person repeatedly responds that they've seen the dots move up one after the other, and then switch to down, even in cases where the actual sensory information is very close to zero and they should respond randomly. So this is just an anecdote, but it kind of zooms in to this larger phenomenon that I'm going to call choice history bias, which have been known in the literature actually for almost a century. And they've been named various things like choice hysteresis, sequential dependencies, and they kind of allow us a little window, as I would like to call it, as at this source of variability in choices that is really intrinsic and relies from the history of people's own previous choices, even in tasks where that history is really irrelevant, because we construct the tasks in such a way that people should really only use their sensory input. And another way that we can think about these biases, this is more for those of you that are psychophysicists, that if we can map the input-output function between sensory evidence and choices, that it usually looks like this psychometric function that you may have seen, and that depending on people's choice history, they shift their whole decision strategy or their well, this is just another way to kind of look at these biases and think about the way that people shift their decision strategy depending on their choice history. So what I'll talk about today is kind of different studies to zoom in on the kind of algorithmic and then neural bases that underlie these phenomena. Okay, so what do we know very broadly kind of from literature about these effects? So we know from mostly behavioral studies that these choice history biases arise most strongly from really the decisions that people make, rather than for the visual stimuli that they see on the screen, or from the motor response that they use to report their choices. We know that they're modulated by the confidence in these previous decisions, and that they reflect a process of long-lasting accumulation of this decision-relevant information across many trials. And there have been different studies suggesting that this choice history effect can be seen in neural activity in sensory, decision-related, and also motor cortex. But how these different kind of neural activity patterns map onto the specific algorithms or the specific processes that give rise to decision has not yet been so clear. And that's what I will focus on today. So first, I will discuss a study that you can check out in eLife, which asks how this choice history affects the within trial decision dynamics. And we use sequential sampling models to study this. The second part of my talk will discuss some neural data with MEG, pinpointing neural basis of history biases in decision making and linking these back to these types of decision making models from the first study. So all of the figures that I will show are in these two publications. So you can check them out and you won't need to screenshot the talk or so. All right, so in this first project, we started from this Actually, I started from a data set that I had collected for entirely different reasons that I was interested in studying. But when I zoomed into the data, I saw all these history dependencies, even though I had told people when they came into the lab that they should just look at the screen and really only use their sensory information. So I was interested in first seeing how widespread this was across different decision-making tasks. So I kind of gathered together a couple of existing data sets that we had in the lab that used a number of different decision-making tasks. And I'll just briefly overview them, but the specific details are not too crucial. So one was a visual motion task, kind of similar to the one I just showed you as an example, where people judged if a cloud of dot was moving left or right. Another one was another visual motion task, but now with a smaller cloud of dots where they moved up or downwards. Two tasks, again, had a random dot stimuli, but now people didn't judge the direction of the dots, but the coherence. So they had to judge whether two intervals whether the second of two intervals contained stronger or weaker motion than the first. These four had single trial feedback. And I also had two data sets with now yes/no decisions. So instead of choosing left versus right, people had to say, did I see a stimulus there or not? And another one in the auditory domain, where people detected the presence of a sine wave in auditory noise. And these didn't have any feedback. And the advantage here is that we could gather quite a large number of people, almost 200, and over 400,000 trials to work with to kind of really zoom into behavioral models of this process. And in each of these data sets, we observed an interesting pattern, namely that if we simply plotted the repetition probability, so how likely every person is to repeat their previous choice, that we see quite a big spread. So in each of these graphs here, you see each observer as one gray bar, and the mean across each study in these colored symbols. So for instance, in this data set, what's kind of striking is that we see people at either end of the spectrum. Some people tend to repeat their previous choices quite consistently, and others tend to alternate. But if you simply average across the group, you can very easily miss these effects. And that's why I think it's kind of an interesting phenomenon that you really need to kind of know what you're looking for. Because if you look at group averages, you will very often conclude that there's nothing interesting there, which I hope to show you that I think that's not the case. So we then asked, okay, people somehow take some information from their past choices and integrate it into the way that they make these decisions about sensory stimuli. How could this decision bias unfold? And to study this, we used a framework which falls under the general umbrella of sequential sampling models. In this particular case, we used the drift diffusion model, which is one of the most well-known of these models. Could I have a very quick show of hands, or you could type it in the chat, like how familiar you are with this? Perhaps a 0 if you've never heard of this class of models, and a 5 if you have used them yourself, or somewhere in between. Maybe we can even do a poll. I actually don't know if that's possible. See a 4 and a 0. Okay, so there's a few experts in the room, and then a couple of people that haven't used them before. Okay, so as more come in, I'll just briefly give a background. If you would like to hear a little bit more, like please kind of stop me and make me explain a little bit more, because it will become an important part of the rest of the talk. Okay. All right, so in these models, what we assume is that people need to integrate sensory information over time to arrive at a decision. And that's especially true because we present these stimuli that have quite a lot of noise in them. So you can't, make a judgment in one snapshot. So the idea is that this decision relevant information is kind of encoded in sensory cortex and integrated over time into what we call a decision variable on the y-axis. And you can see this integrated decision variable into this evolving trace over time. This process starts at some starting point that we call Z in these models, or Z, which is often assumed to be kind of neutral in between the two choice options. So before you see anything, you don't really know what you're gonna be choosing. And this process then unfolds with a parameter quantified by V, or the drift rate, which determines the speed at which you integrate information for one or the other option. So the higher the drift rate, for instance, if you pay more attention, you can have a higher drift rate and integrate this information more efficiently. This process then unfolds until this integrated decision variable hits one of two bounds, shown here as a and 0. And when you hit the bound, it triggers the execution of a response, which in most of these tasks is simply pressing a button with your one hand or your other hand to indicate, for example, the dots move towards the top of the screen or the bottom of the screen. And what's then nice about these models is that we can use not only the choices that people make, but also how long they take to make those choices. And the response time is then the time that they take to complete this decision is then shown here in these distributions at the top and the bottom. So with both these RT distributions and the choices that people make, we can fit these models to behavioral data from humans or animals and uncover these generative parameters. Any, so far so good? Any unclarities there? Please let me know now or forever hold your silence. Okay. Let's say that we assume that something like this is taking place when people make these sensory guided decisions. How can we then have a choice bias? And this can arise in kind of two distinct ways. The first, and perhaps the most intuitive, is to simply shift the starting point of your integration process towards one of the two bounds. So let's say that you've just chosen to report that the dots are moving upwards and that you like repeating your choices, then you will simply shift the beginning of your decision integration process a little bit closer to that bound. And that will lead to a very specific type of alteration in your reaction time distribution shown in orange. And for those of you that like the quantification of this, you can see that the starting point of this integration process has this added bias term, whereas the integration itself just depends on the stimulus identity, the drift rate, and some temporal evolution plus some noise term. If you don't like the equations, just ignore them, and the graphic should tell you all you need to know. Now, there is another alternative for how we can arrive at choices that are biased in some way, and that is by keeping the starting point neutral, but by shifting the integration bias that we add on to our integrated decision variable at each moment in time. So this indicates that at each moment of our integration process, we take a little bit of sensory evidence, but we also integrate a little bit of bias that could be coming from some other source. And I'll talk maybe at the end a little bit about potential neural sources for this type of bias. This leads to a bias that then increases in magnitude the longer you are in a trial, because you integrate more bias at every millisecond or every relevant time step of your brain kind of unfolding. And this leads to very different reaction time distributions, which allows us to kind of disentangle these two scenarios with the diffusion model. And they will also kind of suggest very different processes for how this may play out in the brain. So what we then did in this study is to fit a model where we allowed both of these parameters to depend on people's previous choices. And this dependence is what we're going to call a history shift. So the history shift in starting point is simply how much does people's starting point depend on their previous trial or the previous decision, actually the choice that they made on the previous trial. And the history shift in drift bias is the same, but for this change in their in this drift bias parameter. We can then ask, okay, we see all this individual variability in people's repetition behavior. Can it be explained by one or maybe both of these more mechanistic parameters? And when we first look at the history shift in starting point, we see that it's pretty much unrelated to people's tendency to repeat their choices. But that the history shift in drift bias really pretty strongly captures the degree to which people repeat their subsequent choices. And we were also quite pleased that this pattern seemed to be quite robust across all these 6 different data sets. So here you see that, again, these like visual motion tasks, or auditory tasks, visual contrast tasks. So we think that this captures something that is really goes beyond the specifics of each of these decision-making setups. So in this first part, I've shown you that humans display these idiosyncratic, these highly individual choice history biases across a range of perceptual decision-making tasks, and that these biases are dominated by a history-dependent change in the rate of evidence accumulation rather than its starting point. We did a ton more behavioral modeling that I refer you to the eLife paper, or we can talk more about it if people are interested in that. So maybe I'll pause here for a second before moving on. I'll take a bit of water and see if there's any questions about this section.
Hey, I have one question. Do you think that could be an effect of like trial by trial learning in a way?
That's a great question. Yeah, so I think it depends a little bit on what you mean by learning. So actually, one of the things I started out with early in my PhD is perceptual learning. So where you really kind of improve your perceptual sensitivity, and I think that's not really at play here, but it does if you would operationalize learning as some sort of trial by trial adjustment of your decision strategy, then definitely, right? So. But I think it's a little bit of a different learning than, for instance, reinforcement learning, because we do see that these effects can be relatively independent of whether the choice was actually correct or not. So some people actually just repeat their choices independently of that. So it's some, I would say trial by trial updating process, and you can definitely call it learning, but it doesn't map like super clearly onto, more known types of kind of supervised learning, let's say. Just maybe a sort of internal learning signal that you should stick with your choices for internal consistency or so, but definitely, yeah. Any other questions about this part? Okay, then I will move on and go to perhaps more of the meat of the story here, is where, as a neuroscientist, and perhaps many of you are in the audience, or maybe not, share this similar interest in understanding a little bit better what the neural basis of these processes may be. So if we think about potential neural sources for these biases, we can first start with our understanding of how these types of decisions evolve in the brain in general. Excuse me, I have a little bit of a cough, just 1 second. All right, my apologies. This is always the winter flu season, even without COVID. It's always tricky. Right, so if we think about the way that this type of decision process could unfold in a brain, we normally, pinpoint the encoding of sensory evidence that needs to be integrated in these areas of visual cortex, because in most of these studies we use visual stimuli. And we know that this information is then integrated over time into this decision variable that we can see unfold in parietal and frontal areas. And we also know that simultaneously with this integration process, that the action plan can be read out from motor cortex as people are preparing basically to press a button to report their decision. So in which of these different stages could we then see these types of choice history effects play out? Now, there's of course a couple of different scenarios. And what we did is we designed a specific task, one that I already showed you in one of these 6 data sets, to specifically tap into these neural proxies that we could very easily then extract from our data.
Dr. Urai, if you're having a hard time like speaking, like if you need to, take a brief break.
Okay, I'll just drink a bit of water. I have a bit of a cough recently, yeah.
Yeah, please take your time.
Hmm. Okay. Okay, so we have these random dot stimuli that I showed you at the beginning. And now instead of asking people to judge if they're moving up or down or left or right, as is very often done in primate neurophysiology, we present first a reference stimulus where the motion is always going in the same direction, but moving in a consistent direction, but at a consistent coherence. So 70% of the dots are moving in this same direction. And after a short delay, we show people another stimulus, but now the motion is either more strongly coherent. So let's say 80% goes in the same direction versus more weakly. Where was I? Okay, we have this task where we ask people to distinguish these two intervals. And the big advantage here is that we know from really nice MEG work from Tobias Donner and Marcus Siegel Toby that I work with, is that the stronger you make these coherent motion displays, the stronger the gamma band response over visual cortex. So we have a kind of nice single trial neural proxy for the decision relevant evidence that people need to use to make their decision. Right, so we then also ask for their response where they use either their left or their right hand, which means that we can use the lateralization over motor cortical areas. Again, using this with MEG, which I'm not sure I mentioned, to track their evolving action plan as they're engaging in each decision. So as for the first part of that, let me show you what I mean by this neural kind of oscillatory proxy for the sensory information in this task. Now, this is a time frequency kind of representation of the MEG data that we collected. Again, I'm not so sure how familiar everyone is with these displays, but basically, people sit in this big helmet. We measure this this electrophysiological data, and here on this little circular plot, it is as if we're looking at the top of somebody's head with the ears on the side and the nose here on the front. And you can see that during the time that we present either the strong motion versus weak motion, that there is a large difference in the response over the back of their head, of course, where visual cortex is. And that is specifically in two frequency ranges. So here's the window where we're presenting either the stronger or the weaker motion. And you see that the stronger motion elicits more coherent activity in the gamma range, from 65 to 95 hertz, shown in red, and also desynchronization in the alpha range around 10 hertz. So that's here in these two windows. So this is really an effect of the image that we play on the screen, the video. We can then also zoom in and go beyond the fact that this is like somewhere over the back of the head and use people's individual MRI combined with a linear beamformer to pinpoint these specific regions of interest that we know are involved in these sensory-guided decision-making tasks. And here we specifically use several regions from known kind of atlases of retinotopically organized visual field maps. So here in kind of going from early V1 all the way up to parietal cortex and interparietal sulcus, as well as several motor-related areas. And we can then again kind of just check as a little sanity check that at the time that we present the first reference interval, which is always at 70% coherence, we see this nice boost in gamma band response. And at the moment that we vary the coherence in the second interval, that this coherence then also drives a differential response in an area like V3A and B, which is this known human motion selective bit of cortex. And what's then nice about this kind of this way of analyzing the data is that we can build a GLM model, pretty much a regression model, where now for each area, so going from V1 all the way to motor cortex, and for each of these frequency bands, so we made the selection into the gamma and the alpha band, we can simply ask how big is the effect across the group of this, the category of the current stimulus. Is it the strong motion or weak motion stimulus? And we see that this is very strongly present in kind of early visual cortex and in areas like MT, which is known for motion processing in the gamma range, and that we see this opposite desynchronization in the alpha range. And because we have this GLM framework, it's now really easy to just add another predictor. So to not just ask what is the effect of the current stimulus, but also what is the effect of the previous response on this neural activity. And here we saw something that we thought was really interesting, is that in kind of two specific areas and frequency bands, we see this additional effect, not just of the sensory input, but of the previous choice that people made. Firstly, in the gamma range in this area, IPS2-3, all the way at the top of this parietal set of ROIs that we picked, as well as in the alpha band in IPS 0 and 1, kind of one parietal area lower. So we thought this was kind of a great first, two kind of potential sources of this history bias that we see in behavior. We were also interested in thinking a little bit about how these two might be different, or that they just, that it's just happens to be both in the alpha and the gamma band. And one way in which they are different is that this gamma band effect in IPS2 and 3, here shown where my cursor is only present after people make a correct choice, whereas this alpha activity, this reflection of the previous choice in the alpha band, is only present after people make an error choice. So this goes back a little bit to the question about learning, that there do seem to be differences in these signatures in the degree to which they depend on the feedback that people got on the previous trial, where they presumably have some awareness of, okay, if it was wrong what I just did, that would give you a different update for the next trial on what to do with that information about the choice that you have just made. And these two signals were also a little bit different in their kind of temporal dynamics. So this effect of previous choices in this IPS2-3 gamma is very sustained, as you can see here at the top. So this is the effect of the choice on the last trial during the current trial. And you can see that already at the beginning, there's this effect which kind of sits on top of the current sensory information, which simply rides and is then strongest here when we present the sensory evidence. Whereas in the IPS01 alpha band, we see a more kind of fluctuating dynamic, where this representation of the previous choice is present at the beginning of the trial, then shortly disappears, and then right before we have the second relevant stimulus come up, it's kind of reignited. And this is reminiscent of some other findings in the literature as well, where people find that this history effect can kind of disappear from electrophysiological data and then kind of seems to be reactivated somehow when people are again engaging in the decision that they that they set out to do. So going back to the task, we also were interested in effects on the motor end of the spectrum, because also previous reports have suggested that it's really a bias in the motor system, where you press your right hand once, and this may then have a consequence for the readiness of your motor system to either press that same button again, or maybe you have sort of inhibition where you're actually less likely to come back. And specifically, one paper from Marcus Siegel's lab has suggested that the motor cortex actually induces some sort of alternation, where you have a kind of inhibition of returning to the same motor action. And so what we did in this task is that we counterbalance the stimulus response mapping between individuals. So some people pressed stronger motion with their right hand and others with their left hand, and that allowed us to dissociate the choice history from their action history. And some kind of well-known patterns that we then see in the MEG data. Here again, we're looking at the top of the head, and we see that around the time of the motor preparation, so right before people press the button, we see this lateralization over the strips of motor cortex, but here very broadly across the head, and that is in the beta bands. So well known here is this kind of desynchronization that precedes the actual reporting of the button press. And interestingly, this beta band signal then reverses. You can see that here going from blue to red. And this is called the beta rebound. So this kind of lateralization preparation signal then reverses in this very long-lasting beta effect. And crucially, you can see that here kind of it extends towards the end of the trial, and it in fact remains until the beginning of the next trial. So we see this kind of carryover from the motor response that people give. We can again just check and do our same kind of GLM style approach, where we can confirm that in the beta band, we see at the beginning, so at the reference interval, at the beginning of the next trial, that we see this representation of the last action that people make in these motor areas, like M1 motor area, but also kind of premotor areas and some areas of AIPS. And this was again specific to those previous actions that were correct. So we see here similarity with this parietal gamma band signal. So to sum up, this kind of little intermediate, we have these three cortical history signals. Signals in IPS2-3 in the gamma range and in the alpha range, but in IPS01, which both reflect the history of people's choices, but have slightly different dynamics, as well as a history signal which is more related to action history in these motor cortical areas. And for each of these, that was kind of a nice starting point, but we're then interested in understanding better how they actually relate to behavior. So we then took three strategies to pinpoint more of their functional role. First, we asked how they bias evidence accumulation, coming back to these models that I described in the beginning of the talk. We asked whether they actually predict people's ongoing choice sequences. And I'll end with some ideas about how they reflect these individual differences in behavior that we've seen, again, in the earlier project, are quite often widespread. So for the first question, we again use these two scenarios to understand better whether each of these neural signals might be involved in different biasing effects in evidence integration. So we have the shift in starting point on the one hand, which I'll show at the top, versus a shift in the drift bias. And what we then did is use a type of model where we can allow the single trial neural signals that we have from the MEG to inform the variability in these parameters. So we can simply ask, does each of these candidate neural signals actually predict the shifts either in starting point or in drift bias that we see in behavior? And for the parietal gamma band signal here in IPS 2 and 3, we saw that there was no significant group effects. These are posterior probabilities from a Bayesian model. So the regression coefficient is here on the x-axis. We saw no group effect on the starting point, but we saw a significant positive effect on the drift bias, indicating that this parietal gamma band signal pushes people towards repeating their previous choices. We found no significant group effect for this alpha signature, but interestingly, the motor beta lateralization showed a very different kind of computational effect on the decision process, namely that it had a negative effect on the trial by trial starting point, as if there's somehow a trade-off with this motor signature kind of shifting the starting point away from the previously chosen option towards alternation, which is in line with previous work, whereas the parietal signal seems to add this kind of time-dependent drift bias, which pushes the decision in the direction of repetition. So that's all nice, but of course, with this DDM, it's hard to say anything about really specific kind of causal role in these neural signals and explaining some of the variances that we see in people's choices. So here we took a slightly different statistical approach of mediation, where we can say, okay, we know that there's some effect of previous choices onto current choices. Do these neural signals that we measure actually explain some of this carryover? So we can do that by then taking our trial by trial effects of, for instance, this parietal gamma band signal. We can add a covariate for the stimulus, which we know drives choices, but also the neural signals. And then the way this mediation works is that it's really a series of linked regressions, where the regression coefficients for this, what is called this first path, can be multiplied by the second path to give us what's called this indirect effect. So it's the effect of previous choices onto current choices that is mediated by this neural signal. We can simply add in these two other as well in one model. And what we have then remaining is called the direct effect, and it's kind of the residual variance that we cannot explain with these neural data. So with this model, we looked again at the group, and what we saw was that there is a significant indirect effect of this parietal gamma band signal, but that the alpha and motor beta signals had no effect on predicting these choice sequences. And this is quite interesting, and I'm happy to talk more about this if there's question time, that this motor beta effect did seem to affect starting points, but these effects on starting points may be so small and really, only kind of dominated by these larger effects on drift bias, that they don't really have reached enough power to tell us anything about predicting people's ultimate choices. So we really need these reaction time distributions to pull out this kind of more subtle effect of these motor beta lateralization signals. And lastly, we also see a positive direct effect, which makes sense because with these MEG measurements, we of course don't capture all the neural variability that you would need to really explain the choice behavior fully. And lastly, I want to come back to this phenomenon that I described at the very beginning, where even though the sequences in the stimuli that we presented, which are shown here, were pretty much uncorrelated. So the probability that individual stimuli were repetitions was around 0.5. But individual people in their choices tended to be much more structured. So we see that some people here, we call them repeaters, who have these positive repetition tendencies, and people called alternators have tendencies to switch their choices from one trial to the next. So we were also interested in going back to this and seeing if we could really understand some of this behavioral individual variability, by finding some of these neural correlates. And what we saw is that specifically this IPS23 gamma, which at the group level could predict choices, was only significant in those people that were repeaters. So it seems that this choice history representation is really kind of unique to those whose behavior is then dominated towards choice repetition, whereas this motor beta effect is kind of similar between these two subgroups. Okay, so wrapping up, we saw that choice and action history biases are reflected in parietal and motor cortical dynamics, but with different kind of computational signatures. So motor beta lateralization predicts the starting point of the accumulation process, and parietal gamma band power predicts the drift bias of the accumulation process. It also mediates choice repetition effects, and it is stronger in those people who repeat their choices. And I wanted to
add something.
Of course, yes.
Sorry, slightly technical actually, because I'm working on something similar. And I was just wondering, when you're talking about repeaters and alternators, are you talking about the repetition probability as in like real probability over the experiment in terms of like whether they repeat or not in a given trial? Or is that just like the bias that you calculate using shift in the psychophysical function?
In this case, it's the former. So in this experiment, we don't really have psychometric functions because there's only one level of difficulty. So then you can't fit the sigmoid. I mean, you can, but it's not so meaningful. So in this case, it's really the most kind of simple metric where it's really just how often they repeat. I did, and I don't have those. Maybe if there's time for the Q&A, I can pull it up. I was making some figures where we can fit these logistic models and account for them by having kind of lapses. There's a paper which maybe you're aware of by Ingo Freund, where they have this nice kind of modeling framework for capturing these shifts. And that those kind of weights that you then get for the choice history correlate pretty highly with these very stupid kind of repetition sequences. So for now, I'm using the very simple metric, but I think it would be, it's pretty highly correlated with the more informed model-based metrics. Yeah.
Thanks.
remind me to show you that the figure. It's not in the slides yet. I was just making it for the revision of the paper, so it's a good reminder to add it in. Let me just maybe do one conclusion slide, and then we can discuss a little bit more. So a few things I just wanted to mention is that I think it's really exciting that we find this kind of parietal locus, which seems to be most involved in these biases, because several studies in rodents find very similar things. So both in mice by Yun Huang, here there's two nice papers, and a paper from Athena Akrami in rats, show that if you inactivate an area, which of course is not identical, but it's kind of the analogous area, which is called posterior parietal cortex, if you inactivate it, it actually reduces choice history bias. And in several of these tasks, that can actually improve animals' performance, because these history dependencies can be damaging to performance in case the trials are actually independent. And there are some details there where this seemed to only work well if you actually inactivate in between trials, which is still slightly inconsistent with the more sustained time course that we find in the MEG. So that would be something really interesting to address, in the future with TMS, for instance. I also want to talk a little bit about a more, even slightly more kind of small scale model of how we really think that these different neural populations play out, and how this history bias really adds on to the decision process. There's a few scenarios. So when I started out with this project, I thought it would be really interesting if we see already a change in really the sensory encoding of the stimuli that is affected by choice history. And you can, for example, think about that in this model where you have two accumulating populations that somehow compete with one another, and that they have a starting point, but that they also receive differential input from these sensory populations. But we didn't really find evidence there. So we didn't find an effect kind of in early visual cortex. And also these IPS regions are actually the first that fail to have a representation of the stimulus category. So it's really the sensory information is kind of represented up until there, and then we see this choice history come in. So the way I think about it now is that there could be kind of different sources to these accumulating populations, some sensory, but also this kind of concurrent, maybe it's from hippocampus or from striatum, like these biasing factors that are accumulated alongside, and that then lead to these drift biases. Right, and I think that's the more likely with considering these data, given that this is the first kind of non-sensory region. Okay, this is kind of my wrap-up slide. So we see that these history biases are dominated by the rate of evidence accumulation, and that this is neurally reflected mostly in these parietal and cortical dynamics. With that, I will thank you, and I will briefly thank a lot of the people that I've worked with over the years. Here, mostly the people in Hamburg, where I did my PhD, Tobias Donner, and also Jan Willem de Gea and Constantino Setsos, who I work with on the behavioral modeling project. I haven't really talked about my postdoc work, which was in mice, and kind of doing recordings and really going into the neural data there, but just thanking my postdoc advisor, Anne Churchland, and Anoop Kanal, who helped me with a lot of the mouse work, and also people from the International Brain Lab, a collaboration that I worked with to standardize decision-making experiments in mice as a postdoc. And so perhaps if there's more time after, I'm happy to elaborate on that work a little bit more. And thank you for being here, and I look forward to hearing your questions.
Everyone, so since we have like a more interactive environment right now, we can just jump into the questions, and you can basically raise your hand if you want, or you can just like
you also just speak up, or
yeah, you can just speak up if you like. You can share your video cameras and like
yeah, that would be nice.
Yourself.
Maybe in the meantime, I find that figure that I promised Kai while people are forming their thoughts.
Maybe instead of a question, I can just start, I don't know, rambling a little bit. I was
Yes,
just wondering.
please ramble, and then I will talk about these other things.
Exactly. I was just wondering. So our group is also. So I work with Philip Sterzer. So actually
Okay,
with
yeah.
Anke.
Oh yeah, so
so
you know a lot about this, yeah.
Exactly. It's actually I'm reanalyzing your data from your Nature Communications publication. But anyways, so we're mostly interested in like predictive coding in our group, and so we're especially interested in serial dependence because kind of it's like a nice model since it's integration of prior information with like, updates. And so I was wondering whether you could just maybe comment a little bit on like whether that's something as a framework that's interesting to your research, and whether that like how this maybe plays into your findings, especially the neural signals. And there's some, I don't know,
yeah, I mean, I think you've pinpointed a kind of a tricky thing in this literature where there's a lot of the work from the Fisher and Whitney, like the Nature Neuroscience paper, where they look at these serial dependencies in these angular judgments, right? This was from a couple of years ago. And they always really look at the sensory level. So it's really, kind of more low level. And they often get these repulsive effects, where the idea is actually that if you think about predictive coding, then you want to kind of sparsify your representations, and you don't want to over-represent the same thing, right? So you want to be more sensitive to changes in the visual world. And I think it's quite interesting that we have these kind of multiple things that sit on top of each other. So there's these low-level sensory repulsive effects, which may really be of benefit indeed in kind of the sensory representations in this predictive coding framework. But then on the decision side, so really the choice you make about that sensory information then tends to be at the group level more attractive. And Matthias Fritsche has a nice paper on this, I think current biology paper, on this dissociation. So I think, it depends a little bit if you're more interested in the low level, sensory cortex, where there could be these repulsive effects playing out, or more at the level of the decision circuitry, where there's presumably more of a RL type learning going on, which I, in my mind, haven't quite matched yet with these ideas about the more sensory processing. I'd be interested to hear what you think about that. So of course, with Anke, like the one paper we did, that she did in Journal of Neuroscience, actually doing these regressions, you see that the decisions have a much larger effect than the stimuli themselves, right? So yeah, but I'm also not really a predictive coding expert, so maybe you disagree with me.
No, I agree. It's interesting to hear what you think about it.
Yeah. Maybe I show you one thing, which is here. So for those of you, that may not be so infinitely familiar, like we're diving into some technical details here, so feel free to kind of interrupt if you want to say something more general. But there's these ways of fitting these psychometric models, a little bit like the slide I showed here, where, if you assume that there's this input-output relationship, that you have explicit models that quantify this shift here with the little arrow, like how much do people shift that psychometric function around depending on their previous choice. And so the question was a little bit like, if you do that in this more quantitative way, how related is that to this measure of individual repetition behavior that I use? And so that is here, which I was not going to, as you can see, was not going to put in the paper, but that if you quantify the choice weight, as you call it, that it correlates like pretty highly with this more simple repetition behavior. So you can see that here in this graph, and then the triangles are the people I call repeaters, and the little circles are the alternators. And if you instead quantify how much people repeat their previous stimulus, so that's a more kind of low level effect, perhaps, it doesn't correlate with this repetition behavior. So we're capturing really more of a response level metric. Yeah, I mean, there's more stuff here, but I want to be mindful of not overwhelming everybody with, unless there's interest in that.
Hi, I have another question. Did you analyze
work in Hamburg?
Yeah, we know each other from Hamburg.
Yes, okay. I thought so, but you look different with the glasses. Hi.
Yeah, it's true. I'm on call, so that's why. Yeah, I was wondering, did you analyze the subgroup of tasks that didn't give feedback? Was there would feedback influence the repetition?
Yes, I didn't show it to you, but we did. I will put it here. Yes, great question. This is again coming back to this idea of kind of reinforcement learning or similar processes. So this is again just the behavioral models, because in the MEG data we did always give feedback. But so here I'm showing again the same graph where you have these individual points, right? And you have this history shift that you quantify now separately after correct and error trial. So in this case, the errors are red. And at the top are the data sets with the single trial feedback, and at the bottom are the two without. So I think the course kind of pattern that there is this correlation with the drift bias and not with the starting point still holds in both cases. So this kind of, the fact that most of this individual variability is captured by the drift bias is still there. We do see that it's the shift in the drift bias is even a little bit bigger after error. So there might be in these data sets even a case where if you've made a wrong choice, but you're not being told about it, perhaps there is, there could be like different processes going on. And I think that's a really interesting avenue for really also formalizing then what goes on. And there's some work, Armin Luck has a nice paper in eLife where they also study the role of then specific confidence signals. So like how strong was the stimulus and what was your confidence about it? And of course, making an error could be a proxy for being less confident about your choice. And how does that then impact the way that you update your choice bias later? Yeah. But at least we didn't see a kind of different DDM mechanism of, arising after these two types of trials.
Anyone else?
Yeah, just a quick question. Could you please provide the name of the paper that you just referenced?
Which one? From Armin?
Yeah, yeah.
Okay. You know what? Why don't you email me? And then I can send you a couple.
Sounds good.
Is that easier? Because I feel like otherwise.
Yeah, that would be good. Yeah.
If you just Google me, it should be pretty easy.
Okay.
Yeah, remind me. There's also that way I can put a couple of things.
Yeah, great.
I have actually also found it in the meantime. So this is the paper on confidence that I talked about. It's in eLife from Armin Luck. He's at Oxford now. So you can check out his work. And the paper that I was just discussing with Kai that I think well is this one by Ingo Frind in the Journal of Vision, which is quantifying the effect of intertrial dependence. It should also be referenced in the introduction to my own work. Yeah. And I can send these to you. Great to have so many experts on this here in the audience.
Any other questions?
If no one wants to ask questions, I'm happy to go ahead. I'm wondering, on a bigger picture, so what do you think is the underlying of these differences in choice? Do you think there are people, why are some people more prone to repeating than others? Is that some network thing, or is it maybe, so where does it come from? What do you think? It's speculating, right? But
yeah, so I think kind of more computationally, there's been some modeling work where if you make kind of inferences about these, sequential decisions where you have to make all the choices, that you can capture these individual differences with this subjective hazard rate. So it's kind of the individual estimate of how changing the environment is, right? So if you think that the environment is very stable, then it also makes more sense to have repetitive biases Whereas if you think the environment changes a lot, then it makes more sense to have these alternating biases. But that's still, maybe not so high level in the sense, like, where does that then come from? Right? Why does someone have this positive or negative expected hazard rate? And I think it's still very much open. Like there's been some papers. There's one paper from Justin Gardner's lab, it's a PNAS paper, where they actually link these types of biases to more demographics, right? So that's a little bit of a, I don't know, holy grail, or like it would be really interesting if this actually reflects some real life interesting behavior that matters for the real world. I think in that particular paper, they showed that like people that were, I think, working on a PhD or completed a PhD persisted more after errors. There was some sort of interesting, maybe suggestive pattern, but it was very post hoc and not so highly powered. I must say that in my own work, we have done some questionnaires of different, kind of clinical subtypes. I think you know much more about it, and we never really found a convincing correlation. So what really gives rise to this, it could just be that either people have some more recent experience that they bring to the lab, or it's really something in their life experience that just maybe they have a more impulsive personality or something along those lines. It is, and that's perhaps interesting, people in this study came actually for 2 sessions, which were a couple of weeks apart, and we find like a not super high, but a highly significant correlation between this repetition bias. So it is not some individual effect that is like hugely repetitive the one day and then highly alternating the other time. So I do think there's something about this individual tendency. Yeah. But I don't really know where it comes from.
Yeah, I was wondering if it might, like, if you think of all the work on bistable percepts, people have like kind of an internal switching rate. And if that might be also something that's, although you see it like in parietal cortex, it's somewhere like in the system. That's why. Yeah.
I mean, yeah, now that you think, so I don't, with that, is there clear kind of, has anyone found a clear neural source of that differences, like dopamine level?
Not that I know.
Yeah. I think that there's some suggestions that maybe in autism this is also different. I don't know that literature so well, but there is one paper in mice, and that's perhaps going in an interesting direction, where they actually traced neurons that projected from PPC either to striatum or to motor cortex, and that found that if they specifically inactivated those neurons from parietal cortex to the striatum, that these choice history biases changed. Whereas those neurons from parietal to motor cortex, if they inactivated those with optogenetics, they saw these intact choice history biases, but it changed really in the motor dynamics. So the mice had to move a joystick or so. So ultimately it could really just be neuroanatomy, that maybe there's stronger projections from parietal cortex to striatum in some people that biases them more towards these types of behaviors, if you want to go really low level, but that's kind of very much speculation at this point.
Any other questions? Going once, going twice. I would guess that's all. Thank you so much, Dr. Urai, for being with us today. Thank you so much for the interactive environment you provided us. And like I really like what happened here, like the interaction of academics and postdocs and students here. That's one of our main aims, to basically just create this friendly and interactive environment of scientists. So thank you so much.
Yeah, it was great to hear all your questions. I apologize for my my cough issues. I hope it wasn't too disruptive. Yeah, and get in touch. I'm on Twitter or on email if you want to chat more about this. Yeah, and it was great to hear your thoughts. Thank you.
Thank you.
See you.
Bye. Have a nice day, everyone.